Good evening, everyone. I hope you are fine and doing great. Before starting this session 16, I have one question to all of you. Can you tell me what distinguishes a mathematical model from a poem, a song, a portrait, or any other kind of model? Well, the answer of this question is mathematical model is an image or picture of reality painted with logical symbols instead of with words, sounds, or watercolors. And when these mathematical symbols combined with biology, it creates a boom of computational biology and the dynamic in models we can see in our research area today we are using. And in order to learn about these models, today we are having the guest of the day who are going to give us the ray of information over the same. Hi everyone, once again, this is Kashma Sharma. I welcome all of you in our Bioanalytics and Skill Development Forum session 16. And today the guest we are having, I would like to honor and take a privilege to introduce him. Well, he is a PhD scholar working in the field of computational biology in BSBE department IIT Guwahati. He also worked upon building the mathematical and physics-based models for understanding complex biological events, as well as he also performed the work over the numerical simulation as well as involving different kind of programming languages. I hope most of you are aware about the Julia, MATLAB, and Python. If you're not so, then you will learn today. So please welcome our speaker and the guest of the day, Mr. Shreeman Auromahima, the PhD scholar of BSV IIT Guwahati. Please welcome. Thank you for being with us today. We are very privileged to have you,
Thank you for
Shreeman.
the intro.
Welcome. Now you can start. And I request all the my viewers who are going to learn the same today, please stay tuned with us. This session will going to be very interesting. Thank you so much.
Hello. Yeah, it's visible, right? Okay. So today we'll discuss about mathematical models in biology. So what are mathematical models? Mathematical models are just basically a set of mathematical constructs, like you can say equations or set of equations to study a systems or phenomena. And these equations are often based on some experimental data or some observation, or sometimes it is purely like phenomenological. That means that we have a intuition of how a process or how a biological process is happening. Then based on that intuition, we build a abstract model. Abstract model means we don't capture each and every details what is happening in biology. As you know, most of the biological processes are very complex, and the parameters and variables involved in each processes we cannot measure everything. So we try to build some simple models by taking concepts from physics, specifically from statistical mechanics and thermodynamics, and some parts of math like calculus, linear algebra, probability, and graph theory. How it works, it complements our molecular biology and biochemistry experiment by proposing new hypotheses, or it helps in validating some experimental observation. One thing we have to know that these models are never the reality in 100%. So the two types of system, one is simple system and another is complex system. So complex systems are basically, okay, thank you, systems that have interacting components. Like you know, in biology, tissue is composed of many kinds of different kinds of cells basically, and uh, they are not like static. They interact with each other through secreting some molecules or applying through some mechanical force. And inside the cells also, all the like organelles and all the molecules, they interact with each other, and that's how like living organisms operate. So we want to study, like a system biology help us to study the behavior and dynamics of this complex system. Dynamics means basically how the system is changing. It may be with respect to time, or it may be with respect to space. So why mathematical modeling? Basically, this is how we integrate the systems biology in our cycle of research. Suppose imagine that you did some wet lab experiment, like some common experiment everyone will be aware of. Suppose you did some omics-based experiment and you found a set of genes or proteins, and you think those are important, and you did some analysis, and you have they are like networks. But the problem is, as you know, that suppose you have a large network, right? And you don't know the dynamics of that network. Like if I will ask you, one suppose you just know that okay, this gene activates that gene maybe replaces that other gene, but actually we don't know. If I'll ask what is the rate of that reaction, or how, like if that is happening as a whole, how the cells or systems are evolving with respect to time. Those things most of the time, as it is like we can't measure everything of the system. It is very complicated to measure everything of the system. We can just measure some few things. So it is difficult to answer these particular questions. So what we do, we take those data and we build some hypothesis. Like we can say that one of the most common hypothesis how it is approached, that suppose we have a large network of proteins or genes, we can say that this network consists of some modules. Like you may be aware of, like modules are like if you have a positive feedback. Suppose one gene is controlling another gene, or if you have a negative feedback like that. So we can pick apart some of the parts, and then that small part to understand as a whole how the system is behaving. Then we will propose some hypothesis to test, and those hypotheses can be tested using again some wet lab experiment, and the cycle goes on. So there are different types of mathematical modelings. Basically, this is a graph of the computational cost and spatial details. So at the top, you can see we have molecular dynamics simulation and Brownian dynamics simulation. These are single particle tracking. Like we are tracking the what is happening at each time step to a single particle. But what happens, as we all know, the computational cost of these kind of models are very high. And the another thing is we will not get that much spatial details. Spatial means in the space how like system behaves. There we are just getting that time thing we are getting, like with respect to time how the system is behaving, but we are not getting that much information about the spatial thing. Then we come to little bit this side, like special detail cell. There is agent-based modeling. In agent-based modeling, basically, we'll discuss later on how agent-based modeling work. This is kind of at the sweet spot. It is not too computationally expensive like MD simulations, but at the same time, it gives us some spatial details, and the time scale is very large, and in the scale of how biological processes occurs. Then we have basically reaction diffusion master equation and chemical master equation. These are they're taken to stochasticity in consideration, such that, as you know, like many biological reactions are not deterministic. They are stochastic. Stochastic in the sense that it is if this time step something is happening, in the next time step it is we cannot say deterministically what is happening. The probability part comes. So to model these kind of things, we have reaction diffusion master equation and chemical master equation. Then at this side we have OD and PD, which are basically deterministic model. If our system is we can assume that at least in some part our system is deterministic, then we can use ordinary differential equations and partial differential equations. So let's see some of the basic thing. So what happens in one part of the majority part of the mathematical modeling deals with differential equations, and mostly there are three kinds of differential equation: ordinary differential equation, partial differential equation, and then we have stochastic differential equation. So what ordinary differential equation is, basically, I think we have all studied, simple example like dx by dt is equal to kx, suppose. And here, basically, we have only one independent variable. And that independent variable, when we are modeling biological system, it is mostly time. So basically, we are, suppose, measuring X can be anything. X can be the amount of a protein. X can be the rate of a chemical reaction. Anything. We are measuring something with respect to time, how it is behaving. Those are mostly modeled by ordinary differential equation. The second kind of differential equation is partial differential equation. Here we have two independent variables or more than two independent variables. So basically, suppose we want to know, we want to study diffusion. So this is basically fixed second law of diffusion. So we want to study, suppose diffusions means we all know like particles diffuse or a molecule diffuse inside a cell. So we want to know how it is diffusing with respect to time, also how it is diffusing with respect to space, like in which direction it is going. That also we want to know. Then we have to use partial differential equation. The third type of differential equation is stochastic differential equation. So it is basically when our system is stochastic as we discussed. So if it is not deterministic, then we add some like noise term to represent the stochasticity, and then we model the equation, like model the system, so that it will be like it will represent those systems better than these deterministic models. So what happens, like if we see here, sometimes what happens most of the time we have like complicated equation, not like here we have I have shown like dx by dt is equal to x. This is very like redundant example. Then if you study like complex biology or like some complex phenomena, then you will have some complicated equations. So first we have to know, I think you are all aware that what is like what does it mean that to solve a differential equation. Basically we have here like dx by dt is equal to some function of x and t it will be. So we will integrate this, and we will get, when we will integrate this, we'll get x as a function of time. So by using that equation we can know that like how x evolves with respect to time. But many a times what happens that analytically by pen and paper, integrating these equations are difficult. So we have numerical methods. That's where like numerical methods, and it helps us to delve into those things. So one of the such simple method is Euler method. So suppose you are modeling a differential equation based model, and you are not able to integrate it by pen and paper. So what you can do that you can use something like Euler method. What it does that it approximates that the differential equation like dx by dt is approximately equals to del x by del t. So what does that mean? Del x means suppose time t, you are at a x has a certain value, and with certain delta t, you will take some time interval. After certain time interval, what is the value of the x? That is basically our del x, and we have del t. So what we will do at the like end, you can see that we can get the value of x t plus delta t after delta t time where the x is by simply the equation that whatever the right side function is f of x t del t plus x t. So this may not give us the exact analytical solution, but mostly it will give us approximate results, and that are in most cases that is like good enough to get an idea about how our system behaves. There are some other numerical methods also like Ranjakuta methods, which are like Euler method is one of the basic methods. There are other sometimes what happens that you need some better approximation for your system, then you have to use some methods like Ranjakuta method to approximate those things. So let's see a simple model. So this is one of the simplest model how mathematics is used in biology. As we know, suppose imagine that this is a population. The population can be anything. Suppose it is a bacterial population or some like population, anything, any biological population, and it is increasing. The basic assumption it will be, suppose x is the population size, then we can write how it is increasing. So dx by dt is basically the like rate of change of the population, and any common man will assume has that maybe it depends on what is the current population size, because we know that like from it, like population cannot increase from nothing. There has to be something, from that it is increasing. So maybe it is depends on x. Then what we do, we write a constant. Okay, so this population growth model. So now we have this exponential model, but we all know that this means that the population is increasing indefinitely. But in practical case, if you see, in many cases we have a like cap, because maybe the nutrition will like stop, or maybe some competition arises. So at one point, our like we cannot the population cannot grow indefinitely. Then it will be like the bacteria will go and it will grow at all over the place. So at one moment it stops. So what happens then? We modify this. Like once we see that okay, we are doing some experiment and we're observing at certain point it is stopping. Then we try to modify our initial model, this model, to implement to make it a better model. So how do we do that? There basically it comes like logistic model. So here what we have. Basically, here the equation is dx by dt is rx into 1 minus x by k, where k is the carrying capacity. So what we have done that the rate it will not be like exponentially increasing. At certain point, the maximum carrying capacity is that depends on the experiments, or that depends on basically population. So at till certain point, it will increase. Then it will be like it will stop. The growth will stop. So that equation we can represent in the form of a logistic model. And if we take the integration, we'll get this form of x with respect to time. And if we plot this, we'll get this logistic growth model, where k is basically the carrying capacity of the population. So this is like one example how we can like for simple population growth also, we can first we can have a model. Like basically what happens when in mathematical model, when we are formulating a simple model, most of the time it will not capture everything. But as time progresses and as we observe more like closely what is happening in the system, then we modify our model to make it a better model to represent actually what is happening. So let's see how. Suppose now we know that okay, we can write a differential equation based model, and we can like represent some phenomena. But what else we can do? So that we'll understand through another simple model, basically predator prey model. Suppose like we have a number of prey is x and number of predator is y. So what will happen? We know that the rate of change of predator, it will depend on the birth will depend on x, and the death will depend on xy, you can say, because the predator will eat the prey. And similarly, the birth of the predator will depends upon the prey, and the death, which is basically a simple faster decay. So what we can do here also, we can study the system like that. But here you can see one interesting thing is here we have two variable. We have a number of there we had a just x, so one single population. So here it is a interacting system. So for interacting system, we write coupled ODE. These are called coupled ODE because you can see the rate of change of x, it's depending upon y, and the rate of change of y is depending upon x. So we write these are called like systems of ODE. So these systems of ODE we can analyze in different way to study how the populations are behaving with respect to time. One of the most basic analysis of like this kind of system is done. How like where will be the steady state? Steady state is basically like when the population or whatever we are measuring, in this case the population of the prey and predator, they will not change with respect to time. Basically when we have dx by dt is equal to 0 and dy by dt is equal to 0, that means they are not changing. Those are called steady state. Like in the previous logistic case, you can say that when you are reaching carrying capacity, you have reached a steady state. So we can get the steady state by simply putting dx by dt 0 and dy by dt 0. You can solve it and you will get the value that uh, at 0, 0 and 1, 1, we have the steady state. So what we can do with this steady state? We can simulate the system to get a phase portrait. So this is called a phase portrait. Here in x-axis we have x and in y-axis we have y. Basically those two population. And this is a like two-dimensional problem. We have we are y is changing and x is also changing. So in order to show how it is changing with respect to time, we'll need to plot it in like 3 dimension because time will be another axis. So for support a three-dimensional plots, you know that to analyze a three-dimensional plot is very difficult. So what we do, we just take dy by dx. Basically, we can write like dy by dt and divided by dx by dt. So it will be basically come dy by dx, and we can represent that in our two-dimensional thing. So what it represents that how one variable is changing with respect to another variable. And these arrows represent basically those slopes. These are called like phase portrait. So at each point, the arrow represents where the system will move in the next time step. And the red dot, as you can see, at 0, 0 and 1, 1, as we previously saw, these are the steady state. So if you are at the steady state, then you will stay at the steady state. If I am not perturbing the system, what is the perturbing the system means? I am not disturbing the system by any way. Disturbing means I'm not some new prey or predator I am making go into the system or something like unnatural is happening. If that you are not doing as a natural system, it will stay at there. But if you see actually what happens practically, if it is 0, 0, then you can understand that it will stay there because nothing is happening. But if you take that only one prey and one predator is there, actually after some time it will come back to 0, 0 only. But those can be considered as a steady state in this simplified model. But the interesting thing happens at like what is happening around the steady state. So if you can see around the basically 1, 1 steady state, what is happening? The arrows are moving around a circle. So what does this represent? This represents actually oscillation. Oscillation means if you can see what will happen in most of the natural case, if you go to a forest or anything, if you see what happens, suppose now you can if you can see the right side plot, suppose at the beginning the prey number is high, then what will happen? The predator will start to eat the prey. At certain point, the number of prey will decrease. If the number of prey will be decreased, then the predator will get hungry and they will die of starvation, some predator. So the number of predator will decrease. Then what will happen as the number of predator will decrease at that time the number of prey will increase. So this will behave like a oscillations that is represented as this like arrow around like circling around the arrow around the steady state. And these kind of steady states are also called like center text steady state. There are different kinds of steady state which if you are interested, like you can study in any dynamical system textbook, standard textbook, you can see. So what happens that this is basically how we study. Suppose now we know that just from a simple differential equation, we can observe certain behavior, actually what is happening in the natural system by just writing a simple ODE and studying how the ODE behaves. Then we'll move into a different kind of modeling called agent-based modeling. So these are basically we are writing the differential equations, right? In agent-based modeling, what happens? Like we'll represent the components of a system as an agent. Basically, you can imagine, suppose your system is tissue. You want to study a particular tissue. Then the agents will be cells. If you want to study, suppose a human population in a city, then your agents will be humans. If you want to study like a only single cell and how certain molecules are behaving, interacting inside that cell, then your agents will be those molecules. Basically, how like whatever the system is made up of, those are called agents. So what happens in agent-based modeling? We basically take this is basically a bottom-up approach. What we do, we make some rules. Those rules can be basically derived from some first principle of physics, or it can be some simple rule based like predator prey. We assume that the predator number of predator is like depending upon prey, and that too like in a particular simplistic way we wrote the equation. Like that we can assume some rule how something is happening in biology, and we will based on that rule we will evolve our system, like we will make the agents interact with each other and how the system is evolving. Then we will study like at what time point, like which time, where is the system going, and is there any interesting phenomena or not. So in agent based modeling, basically there are two types of agent-based modeling. One is lattice-based and another is off-lattice. So in lattice-based modeling, what we do, basically we represent the agents on a lattice. Lattice is basically a grid. You can represent like in a two-dimensional, you can get a square grid. Here in the picture, it is shown as hexagonal grid. So any kind of grid, or you can make three-dimensional. Like basically two-dimensional are easy. So many models we use two-dimensional, but you can in any dimensional you can use a grid. And on the grid, one grid point you can use at certain agents or cell. And these are some of the example. Like in LGCA, what happens? One like multiple cells are represented by one lattice. So these are helpful. Suppose imagine that you are like there are two different types of populations of cell, and you want to know how they are interacting with each other. Like suppose they are collectively migrating somewhere, or they are doing some interesting thing by interacting with each other. I want to study those things you can do by applying LGCA. You can basically take one population of cell at one lattice and other population at another lattice, and you can make some rule and study those behavior. The second type of thing is called cellular automata. In cellular automata, we basically like each lattice is represented by a single cell, and those cell you can like at each time step you can vary the cell. Like something will happen. Either the cell will support cell is growing, or the cell is dividing, or the cell is moving. According to like your biological problem, you can make some rule and make that happen. The third type of thing is cellular pods model. Here, multiple sites are represented by one cell, and these have certain advantages which we will discuss. The second type of thing is basically off-lattice based. Here we don't have any lattice. Then what we will do? What we will do? We will use like agents. Basically, how we will measure. What we will measure? Suppose we can measure that two cells are there, and we can measure what is the force between the center of them, or we can do like we can track the boundary. Basically, suppose the multiple cells are there, then we can put some vortex on their like boundary, and we can track how they are basically evolving with respect to time. Or we can do like front tracking, where basically the resolution of the boundary is increasing. In central based model, similarly, like for lattice based, we have different kind of like one agent can be for one cell, we can represent one agent. And for multiple cells, sometimes if we require, we can represent one agent also. So these are like different, basically grades of model you can use according to your need. We'll see just one example, that is like cellular pods model. So cellular pods model is one of the lattice-based example, like lattice-based agent-based modeling. So what happens is here it is like physics-based model. So as you know, like it is based on basically statistical mechanics concept. And in statistical mechanics, we have a model called Potts model. A Potts model is basically a, like if you know ferromagnet to paramagnet transition, how it happens. For that transition, there is a model called Potts model, and that model, like from that model, we take some idea and we apply it on biology to make this cellular Potts model. This model like behave mimics how the biological cell moves actually very well. And originally they are developed for cell sorting. Cell sorting is basically when the development happens. If you see like if there are like randomly intermixed different kind of cells are there, as the development happen with at the time like increases, what will happen? Those cells will form different patterns. Like different types of cells will segregate from each other, and they will form different patterns, which basically gives rise to tissue. So one can ask the question, even if we know, like at the molecular level, we know that what molecule is acting on this or what protein is there, but actually how those are giving rise to this kind of emergent pattern, those we can't say from a like reductionist approach, because when we are doing experiment, we are just isolating a cells or isolating a particular molecule and doing experiment. But in biology, what happens in actual living organism? Those cells interact with each other, those molecules interact with each other in a complex way, and then they give rise to some interesting thing that we can capture by this kind of models. So this is the model. So we'll like simply understand what this model is. Here you can see that like this number you can see like 1, 2, 3, 4. These are actually one cells. So we represent cells on the lattice like this, and the zero is basically the extracellular medium. As you know, each cell is surrounded by some kind of matrix or some kind of medium. So what happens? We basically like we have a principle. This is the below equation is called the Hamiltonian. I don't know if you are aware about Hamiltonian or not. Hamiltonian is basically a concept which is used to represent the energy of a system, right? So what happens? We assume that like these are our models assumption. All the mathematical models, the most important thing we have to remember that we assume something either based on the certain theory or from certain experimental data, and then from based on that assumption, we basically build up our models. So here we assume there is a famous theory of cell segregation called differential adhesion hypothesis. So what it says that like in a tissue, if two similar types of cells are nearer to each other, what they will do, they will have like less surface tension between them. So the as a whole, the surface energy of the tissue will be more. But if two different kinds of cells are there, the surface energy between the boundary of the cell are more. So what will happen? As a whole, we assume that the tissue tries to minimize this surface energy, right? So that it will like minimize the energy and it will go to a stable state. So it will go to basically equilibrium. So we write down these kind of rules, what we understand, like there are experiments and there are theories saying that these are one of the major driving force how tissues are formed. So based on that idea, we basically formed this Hamiltonian. So this Hamiltonian basically have the first part represents the cell-cell adhesion, how different cells interact. Basically, if same type of cells are there, that is more favorable. And if basically different types of cells are nearer to each other, then that is unfavorable. So what we will do? We'll move the cell, and these numbers are actually called pixels. So we will not go into that much complication. So what will happen that there is a certain simulations we do called Monte Carlo simulation. By doing that simulation, we like move the cell, and we after sometimes we can see that like how the system is how as a whole the tissue is behaving. The second term in the energy is basically area constant. That basically we have kept that like as a penalty. As we all know that biological cells have a specific area. So what we do that like we give a penalty if the cell's area is like varying too much, then or too little. It doesn't happen actually in biology. So we give a penalty to that so that the cell's area will be more or less at the same place. So then we simulate the system by using this energy term probabilistically. Like how we do that, we basically apply some probabilistic method to simulate the system so that as a whole the tissue will go to global minimum energy. Okay, so this is your like experimental methods. This is not a model, but this is an experimental cell segregation which was done. Like it is very quite old experiment where the scientists what they did, they took some neural like retinal cells and pigmented retinal epithelial cells. You can see like one is light cell and one is dark cell. Then in the like they took the cell and at the beginning they were like randomly intermixed with each other, and as the time progressed, they saw that this particular kind of patterns form. This is basically a cells of a chick embryo. And what happens that one kind of cells actually surrounded the other kind of cell. This is actually experimentally observed. This is a biological experiment. As you can see, the model I just show you, like cellular first model, if we can vary the parameters basically in the no, in the Hamiltonian which we wrote, we can vary different parameters like how cells are interacting, like how this light cell and dark cells are interacting with each other, how light cells are interacting with the extracellular medium. Those things we need to vary. So for a particular set of parameter, we can actually mimic what I showed before in the experiment. Here also you can see that one kind of cell is surrounding the other kind of cell. Now, once it matches with the experimental observation, what we can do that now we can play around it to like change those parameters of our model. Like okay, I will increase the like suppose attraction between different kind of cells and see what happens, or I will decrease the attraction between cell and medium and I will say what happens. Actually, very this is not the only pattern. Many different patterns can arise if we do that. then what will happen? Then maybe suppose we did this, and in biology we observed that okay, in one particular organism that thing is happening. Then we can connect our model that okay, maybe our model is able to explain at least in part how that biological process is happening. Still, it is not able. It will not be able to answer each and everything like what is the rate and all those things. But some like you will get an overall idea that how cell sorting happens in like organism. What are basically the governing principles that is driving this kind of behavior? Because those things you cannot get. Suppose you did this experiment and you observe that that is happening. But why? You cannot take a molecule and you will never find a like single gene or single protein which will give you the answer that okay, this protein is there, that's why all this like pattern and all the patterns are emerging or all this behavior are emerging. So here like we take helps of the like simplified model to explain this kind of behaviors in biology. So if you want to go into this field, basically first you have to know actually different kinds of at least some discipline of math and physics. What I showed in the first slide, basically in math you have to be thorough in calculus, linear algebra, probability and statistics. And graph theory is not mandatory if you want to study like protein network and those kind of network. For that you have to know graph theory, but at least linear algebra and calculus and probability you have to be thorough. And from physics perspective, you have to know statistical mechanics. Statistical mechanics is basically when we study different thermodynamic system from a probabilistic point of view, not from a like coarse grain how thermodynamics study. Those things you have to know. After that, to create this kind of models, like I showed, basically the ODE-based model or agent-based model, what you have to do, you have to write some code. Like you cannot just by pen and paper, it is very difficult to like simulate. If your system is very complex, it is very difficult to get the analytical solution every time. So you will write some code to like make your like you will convert your model to an algorithm. Then you write the codes using some language, and then you will study your system. So mostly the in computational biology, people use like C and Julia because they are very fast, right? What happens? This kind of simulation actually takes time. If you want to study certain large system and if you want to study, if you want to perform different experiments, it takes actually time. As you know, like Python and MATLAB, these are like interpreted language. Although these are easy to learn, but you will like feel that when you are doing actual simulation, if you are studying some actual biological tissue or actual biological chemical reaction or something, and if you are simulating, if your system is very complex and large, then this interpreted language, like although these are easy to write, but they will not be that much fast. So we use C, or you can use Julia. Julia is at least in many cases it is as close to as in terms of speed it is as close to as C, and it is faster than Python and MATLAB in most of the cases. So and the code of Julia is much more like easier to write than C. So you can like now Julia is basically an emerging language which people are using in this computational biology field to create their models. And sometimes you can use some open source toolkit like JSIM. You can discover JSIM is basically like I showed you the creator prey model and those kind of ODE based model. So JSIM is basically for to if you have a differential equation, you can write that and numerically you don't have to solve it. Numerically, your computer will do it, and you can study your system's behavior by using those basically those tools. Unlike computer 3D is there, that is basically used for this kind of cellular post model and agent-based model. But sometimes what will happen once you delve into your particular research area, sometimes these like graphical user interface tools will not be enough for your research purpose. So you have to like learn at least to like how to write code in one of the language in order to properly develop a model and study. Thank you.